And here we are in part 3 of chapter 8, Neurons. We're going to be dealing with the synaptic transmission. This is very much in the same vein as endocrinology or the signal communication that we talked about in chapter 6. It's just going to be applied to the specific situation of neurotransmitters and synapses. So to put together what we've covered so far, up here in the cell body and the dendrites, we have graded potentials that can summate, interfere with each other, or synergize, add up together to create the decision here in the axon-hillock initial segment as to whether to fire off an action potential down the axon. Now, we have transport as well. We've talked about fast axonal transport. We sort of ruled out slow. To get materials to and from these distant presynaptic terminals. And put together, we also have resting membrane potential from start to finish. In fact, resting membrane potential is a part of the dynamics that we're going to talk about here, what we call neurotransmitter and secretion dynamics. That's a mouthful. What we're talking about is whether or not these neurotransmitters get secreted, and it's a vesicular exocytosis. That's as far mechanistically as we're going to get. We are going to show you how we get to the point where this happens. We're not going to break that mechanism out. The response, what's happening in the downstream neurons and or glands, they can be glands or muscles, or even some nearby cells, because response, there is a paracrine, sort of action going on here that isn't necessarily the immediate target, and I'll explain that later. And then this one folks tend to have more trouble with, is this idea of termination. If we have these without this, we have unrestrained signaling. And back to my RAID metaphor on a bug, what you're doing with the active ingredient in RAID is blocking termination of acetylcholine signaling in those bugs, and that's why they pass away. We will talk a little bit about recycling as well, and the reason why we talk about recycling is it has very, very important therapeutic considerations in neuroactive drugs. Your SSRIs, serotonin reuptake inhibitors, SFRIs, a newer generation of those are very, very important. So we want to get through this mechanism so that you can understand what those drugs are really doing. But altogether, this is signal integration. We're making decisions here, we're making decisions presynaptically, and we're making decisions postsynaptically. Is there enough voltage here? Do we have enough action potentials to cause more or less exocytosis here? And the amount of neurotransmitter that's reaching receptors on the downstream cell. Little summary slide. This is good, kind of bringing pieces together. Here's our cell body without dendrites. It, could have dendrites. It's showing the axonal transport. We're using those motor proteins and ATP to do this. This synaptic cleft is really this area right here. So this is the end that we see right here. And what's going on is these vesicles stack up on the surface here. And when the appropriate action potential signal comes along, we release those vesicles and the contents therein. The vesicles don't leave the cell. The vesicles become part of the plasma membrane. But the contents are released and they diffuse to interact with receptors or other enzymes in this cleft. It's entirely possible that this neurotransmitter doesn't make it to receptors, because part of the termination mechanism is to get rid of neurotransmitter. So we communicated synapses, and not all synapses are chemical. Some very, very fast synapses, some of the fastest reflexes in your body, are in fact like gap junctions. They literally have, it's a very, very fast kinetics, so super fast reflexes, often to auditory signals or your blink reflex are all involved these things. They're very rare though. The vast, vast majority are these chemical synapses. And for a moment, put your hands together as if you were in prayer, that sort of prayer pose, and push your hands together. The space between your fingers right now is actually more than the space. I mean, I'm talking about from one hand to another, is actually larger than the space between a presynaptic neuron and the postsynaptic membrane surface. It's a very, very small, and I have some electron microscopy to show you. Actually less than the diameter of a vesicle in some cases. We do have neurotransmitters. These things are molecules that will affect receptors on that downstream surface. The main difference between neurotransmitters and neuromodulators is that neurotransmitters are very, very fast acting. They act in milliseconds. Their half-life is in milliseconds. Neuromodulators cause metabolic changes. If neurotransmitters are primarily ionotropic, if you remember that term, ionotropic receptors, you're getting a quick change in membrane potential here leading to graded potentials. Neuromodulators are longer term. They're more like, they just last longer. They have longer kinetics, and the effect on the membrane, the effect on the voltage of the membrane or the response is longer duration. We're talking about orders of magnitude, seconds, minutes, hours, lifetimes. Neuromodulators and neurotransmitters are similar in how they do their thing, but the neuromodulators are much more durable than neurotransmitters are. The receptors involved in these, we talk about chemically gated or ligand or ligand, however, whatever accent you have, chemically gated ionotropic receptors. This means that the channel is specifically gated to the presence or absence of a molecule, and it is also a channel. So as soon as that receptor is activated, you get immediate and very rapid change in permeability to ions across the membrane. I have seen some real-time recordings of this, and it's in literally fractions of milliseconds. It's open, closed, open, closed. And however long that ligand is present and binding is when those channels are kind of open and closed, but it looks almost like a on-off switch, but it happens super fast and isn't constant. As receptors are interacting on and off with those ligands, they open and close. Metabotropic receptors, on the other hand, are usually G protein coupled receptors. They use more metabolism. They mediate slower responses, but generally more durable responses. We are going to talk about long-term potentiation. Long-term potentiation is mediated by metabotropic receptors. What do you mean long-term? Long periods of time, potentiation, changes in the sensitivity of the membrane. This is where the neuromodulators do their thing. And so here we have the two paradigms. We have the ionotropic receptors. We have the G protein coupled metabotropic receptors, both of which are summarized in chapter 6. Fast change in permeability creates a change in voltage. Voltage sensitive proteins, very fast response. This one can do the same thing, but it can also do other things. Maybe we're doing something else that's causing long-term change in the number of these. So we become more sensitive to that extracellular signaling molecule, or maybe something here that downregulates those, we become less sensitive. Did you know that if in the unfortunate event that a young child doesn't hear a spoken language, that they become permanently disabled and are unable to develop speaking language? That's because these pathways for learning language are a window in a period of time in that child's development. And if we don't have that exposure, you don't sensitize to language. It's a very extraordinary and unfortunate event if it happens to a person. But an ionotropic, those are receptor channels. Metabotropic, those involve metabolism. They involve a second messenger of some kind that may mediate channels, but also may mediate other responses that make longer changes to the membrane. Let's talk about neurotransmitters a little bit. Neurocrines, if you will. That, lots of intermediate words around. Neurocrines, neurotransmitters. I'm not going to use that term very much. Acetylcholine is kind of the king. It's the most common. Your cerebral cortex is dominated by acetylcholine secreting neurons. We call them cholinergic neurons. Alzheimer's is most closely associated with loss of acetylcholine-mediated signaling in central nervous system. We have the amines. They just keep popping up. The human body is elegant and vexing that way, because we're using amine-derived. These are catecholamines, to do things like being in hormones systemically, but also as neurotransmitters. And that's what can make therapy very, very tricky, because you get side effects because these molecules are being used in more ways than one. We also have amino acids proper. So we have derived amines derived from amino acids, and we actually have actual amino acids. This one's called gamma aminobutyric acid, or everybody just calls it GABA. It's a very, very important inhibitory neurotransmitter. Its most common receptor target is a chloride channel, which hyperpolarizes neurons. It's also one that tends to go away in advanced age. So people tend to lose their filter a little bit as they get older. Peptides can be Neurocrines, neurotransmitters. Substance P is involved in pain signaling, but certainly endorphins and enkephalins are known about for their sort of euphoric and good feeling events. Morphines and things tend to mimic these peptides. That's why we call them the opioid peptides. And then lastly, ATP and metabolites of ATP, adenosine monophosphate, can also serve as neurotransmitters. So not necessarily cyclic AMP, this is just straight up linear AMP, but again, they both can serve. ATP is particularly in taste buds, for instance. Then you have some what are kind of considered non-canonical neurotransmitters. They're coming along. They are moving into the neurotransmitter space by leaps and bounds. The gases in particular. So nitric oxide was awarded the Nobel Prize back in the 1990s for being a vasodilator. But some of my work as a postdoc, we were looking at protein modifications. So this is a signaling molecule, and it flies through membranes. It's almost like a radio signal going through membranes. It can go to distant targets relatively quickly, and it works as a neurotransmitter. Carbon monoxide. We often think of having carbon monoxide sensors in our house or in certain activities. Carbon monoxide has been shown to be a neurotransmitter, albeit at very low levels. It has been confirmed as a neurotransmitter. Hydrogen sulfide. If you've ever been to Yellowstone National Park and smelled the hot springs and the geysers there, or if you've gone down to French Lick, Indiana, the Pluto water in the French Lick Resort springs has a fair amount of hydrogen sulfide in it. It smells like rotten eggs. Very, very potent neurotoxin. It interacts with proteins, but at very, very low levels, it functions as a neurotransmitter. And then lipids, and this is clearly the newest category in terms of what's going on. Most common lipid that's been studied or is being studied, because it's way behind everything else, is this one right here, tetrahydrocannabinoid. THC is a lipid that is neuroactive and works as a neurotransmitter. And so research is slowly coming along. It's been it was very lowly, very small studies for many, many years because of its association with being an illegal drug. But research is rapidly ensuing just because of the widespread use now. It is so widespread, we're starting to see side effects that were unanticipated in truly large populations. So getting a handle on THC and its benefits and its negatives is very, very important. Here's that electron microscopy image that I promised I would show you. This is actually a neuromuscular junction. This is muscle here, and those are sarcomeres. But in any case, here is this presynaptic terminal, and you can see how small this space is between the presynaptic terminal and the postsynaptic membrane. Here's the vesicles, super small, even in cellular terms. But that space is teeny tiny at a molecular level. You can even see this little black line, kind of foggy line here. those are probably proteins on the surface that have stained in this, which is absolutely amazing to me. So let's get into the mechanism of neurotransmitter secretion. We're going to do this in the big mech fashion. So we're going to do this step by step with these little circles here. What happens is we have the axon here, pre-synaptic terminal. This action potential is moving down this axon, so it's coming across the whole axon. It's not just one side of the axon. That voltage deflection comes around and hits this region in red right here. This region in red right here has a slightly different constituency of membrane proteins than this area does. This red area not only has the regular action potential proteins, but it also has lots and lots of calcium channels that are voltage sensitive. So that action potential comes along, spreads through, we get a voltage deflection here. That voltage deflection triggers calcium channels. One thing I haven't mentioned about calcium is that calcium is very, very low. I mean 2 parts per billion or less inside the cytosol of a cell. There's a lot more calcium outside the cell than inside the cell, and if the cell contains calcium, it contains it in the endoplasmic reticulum. It sequesters it, it keeps it under lock and key. So when we open these calcium channels in response to the voltage, calcium then will be able to stream in quite rapidly. Yeah, it's going to cause depolarization. We're already depolarized, but in any case, calcium is acting as a cofactor / second message. The calcium will bind with proteins that are involved in exocytosis, the snaps and the snares and that sort of thing. And they will cause migration of vesicles and fusion of vesicles with the surface of that presynaptic terminal using ATP. So calciums are a cofactor. Calciums are also a second message here. Those vesicles merge, the neurotransmitter streams out, and what we get is an increase in concentration of neurotransmitter in the space. We call this the synaptic cleft. It's extracellular space. Okay, so if there are receptors present, and there should be, if there were no receptors, we'd have no response. If there were more receptors, we would be more sensitive to this signal. Okay, keep that in mind. The abundance of receptors can play a role in whether or not we get a downstream signal or how much signal we get relative to the strength of this signal. More neurotransmitter, more strength. The neurotransmitters bind the receptor, and off we go. If it's a muscle, we're going to see a signal transduction event leading to contraction. If it's a neuron, we would see this graded potential ensue. Maybe this is an ionotropic receptor to calcium, or neurotransmitter, excuse me, and that causes an influx of sodium or some other ion causing a graded potential. So we have seen secretion. Now let's bring in termination. Critically important. Just like CO2 is critically important to life, as much as oxygen is in many, many ways. As we are secreting neurotransmitter, that neurotransmitters have millisecond half-lives. I mean, they're super short. They are an endangered species almost as soon as they hit the synapse for 3 primary reasons. Is that we need to turn off this signal almost as soon as we turn it on. If this was a neuromodulator, then the duration might last longer. There's more. I don't want to get too detailed here, but in any case, neurotransmitters have super duper fast kinetics, and so they have to literally disappear in milliseconds, or they're going to be stuck to those receptors, and we're going to continue to get signal. More signal is analogous. More receptor interaction is analogous to stronger signal. If we have more action potentials, we'll get more neurotransmitter, more interaction, stronger signal. If we increase the half-life of neurotransmitter, that's the equivalent of having more neurotransmitter, more interaction, stronger signal. try to save that idea if you can. If you need to listen to that again, back it up 15 seconds and listen to that. More action potentials mean more neurotransmitter. It's in the synapse longer. If we increase the half-life for whatever reason, not by increasing neurotransmitter, but by other reasons, that would be analogous to a stronger signal in biochemical terms in this synapse. So here are the receptors. They do obey kinetics here. As the rise in the L, which is neurotransmitter, with the P, that's the receptor. As we have more L, it will shift this to the right. We'll get more bound. As we get rid of L, we shift this to the left as ligand becomes unbound. This is known as the law of mass action. I have not emphasized this semester, but I want you to understand that that's what's going on. If we add mass over here, some of that mass is going to shift to a bound form. If we subtract mass over here, some of this is going to compensate. Just like if you imagine having an aquarium. Imagine this is an aquarium and we start scooping water from this side. Well, water is going to shift over. Or if we start scooping water from this side, water is going to shift over. That's a really nice metaphor. Or if we add water, let's say we pour some water over here, it's going to rush over and we're going to reach or try to reach equilibrium. That's the binding and unbinding kinetics of law of mass action. So that is why it's important, because it's predicated on how much neurotransmitter we have. The more we have, the more is going to be bound. The less we have, the less it's going to be bound. And their downstream signal there, response. So that's your normal signaling. But how do we stop this? Well, we stop it by getting rid of that neurotransmitter. 3 mechanisms, 3, count them. The first mechanism is collectively known as reuptake. So if we release neurotransmitters, reuptake means we're pulling them back into cells, only not usually through vesicles. Usually it's a carrier of some kind. And we're going to process that neurotransmitter and get it back into a vesicle. If it's going into a glial cell, it's probably just going to be destroyed. But both of these processes are known collectively as reuptake. So neurotransmitters can be returned to the terminals or transported to glial cells. That is reuptake. Here are our receptors down here. I want you to look here at number 2. Number 2 here, and it's a fast flash. Number 2 here, that's an enzyme on the inner surface of this synaptic cleft. That enzyme is tasked with metabolizing neurotransmitter, and they're usually pretty fast. For instance, if these red dots are acetylcholine, this enzyme would be generally known as an acetylcholinesterase. It's going to chop it up into acetate and choline. That's going to contribute to the loss of concentration of acetylcholine, loss of concentration of neurotransmitter. The more enzyme we have, the faster it disappears. So that is known collectively as enzymatic inactivation. So we have reuptake here at 1, and that's going to go on as long as there's a neurotransmitter out here. We have enzymatic inactivation. And the third mechanism is because this synaptic cleft is in fact an open system. It's extracellular space. The third mechanism is straight up diffusion. And this is where some really interesting things can happen, because maybe those neurotransmitters begin affecting things like blood vessels. So if your brain's working real hard, it needs more nutrients. And so a lot of acetylcholine is going to be diffusing out of that synapse if this thing's firing like mad. And that may in turn affect other cells like blood vessels and cause vasodilation. If you've ever seen or had a functional MRI, that's one of the ways you can detect brain activity is blood flow in that area, and it's mediated by neurotransmitters diffusing out of the synapse. If we lose neurotransmitter in any one or all of these ways simultaneously, that's usually what's going on. you have change in the response. We diminish the neurotransmitter, the binding kinetics shift to the left to the unbound form, and response goes away. Okay? These things will literally move off of those receptors. And students traditionally have more difficulty with this than anything else in this particular section. I guarantee you there will be questions on this on the exam. So get to know this mechanism so that you can articulate it properly. Termination is just as important, and I've mentioned this in the past. Neurotoxins most often are inhibitors of neurotransmitter termination. Here's acetylcholine. We have the acetate on the left, like the acetate that you see in the plastics. It's such as a polymer of this. It's acetic acid, it's an unconjugated form of it. And then we have choline, a nitrogen-centered little molecule over here, very lipophilic. Bound together, that's what makes acetylcholine. We're going to show you that the secretion and the termination in acetylcholine. So we're applying this to a specific neurotransmitter this time. You're going to see secretion, the action potential, and then what happens to acetylcholine in the receptor. This has major ramifications for Alzheimer's, major ramifications for neurotoxicity. So acetylcholine is manufactured. It's made from acetyl-CoA. Okay, so acetyl-CoA is a common metabolite made from mitochondrial function, and that's one of the reasons why we use fast axonal transport to bring mitochondria down to this presynaptic terminal. Mitochondria are a very common cargo on the fast axonal transport, and it's wild when you see the microscopy. They look like little subway cars moving down the axons. It's really cool. So acetyl-CoA is manufactured by enzymatic conjugation into acetylcholine. It gets packaged into these vesicles and is ready for secretion. I'm going to hold on for step 2 here. We're going to get our action potential. We're going to get our calcium. The calcium causes exocytosis. And now acetylcholine is out here, and we've got boatloads of it out here. Yeah, it could bind a receptor and mediate signaling if it encounters a receptor. One of the things that happens right away with acetylcholine, if it does encounter a receptor, it's most likely going to encounter an enzyme, acetylcholine esterase. It's going to chop it into acetate and choline, and that's why I showed you that picture before, acetate and choline. So acetate, I don't have a good animation here. Acetate will just diffuse out. So that's the diffusion mechanism. Choline, on the other hand, is brought back into the cell using the sodium symporter. This is secondary active transport, by the way. It's secondary active transport because we use ATP to sustain that sodium gradient, and we're using the sodium going down its gradient to bring choline in against its gradient. Choline's higher inside the cell. So we're going to move choline in, secondary active transport, symporter, okay? And then choline can be recycled onto acetyl-CoA. So this is reuptake. So step 2 is enzymatic inactivation, and that's consistent with the previous figure. In the previous figure, we had this at step 1, but we can't get to here without going through here first, and that's why it's 3 instead of 1. And acetate, we don't have the diffusion. So we have reuptake, enzymatic inactivation, and then we have diffusion of acetate out of the synapse. It's the same story, it's just that we've applied it to acetylcholine. And there's that recycling, that process. We often recycle neurotransmitters because our brains are active, and so it takes a lot of energy to manufacture neurotransmitters. Why not use them again, since their half-lives are generally so short? Now we're going to get to signal integration. We've done the mechanism of signaling here, and I've alluded to some things like, for instance, more action potentials, more neurotransmitter, stronger signal. Longer half-life, more neurotransmitter, stronger signal. That's the basis of signal integration. This is also going to include some neuroanatomical stuff. So, but we're going to start with the action potentials. So if we look at the neuron here, and this is a graded potential up here, I've just superimposed this figure. This is occurring here at the axon hillock initial segment. You see a graded potential and the action potential showing up. It's a modest signal. The signal's bigger over here because it's earlier in its life. As it moves, it gets a little bit weaker, but it's still threshold. We hit minus 55, and when we did, we fired off action potentials. Those travel down the myelin sheath. You don't see any graded potentials, or you see teeny tiny ones. Those are each of the segments firing off. But that's what you're seeing here is action potentials, and that leads to some neurotransmitter release. What about a strong signal? And I want to point something out right away. We see a bigger graded potential. We see a bigger graded potential here, but look at the density of the action potentials. They're closer together. That's because this signal, those action potentials are firing off earlier in the relative refractory period. We have more more action potentials per unit time because the signal is stronger and it's overcoming the refractory, the relative refractory period. You've got elevated voltage, it's stronger, means we're going to fire off action potentials sooner. So look at the previous figure, look at the density of those action potentials. We now have at least, it's hard to see, but at least 8 lines there, whereas in the previous figure we only had 4 in the same unit time. This means more depolarization, more calcium, and more secretion. And again, I've got the figure here, lined up and so forth. Now this figure is really, really interesting to me because it shows the cell body, and these blue ones are pre-synaptic terminals all over this thing. Okay. We also happen to have some glial cell foot processes here. That's fine too. But you can see that if a graded potential weakens over time, so let's say this guy out here fires off, put some neurotransmitter, that graded potential is going to get weaker and weaker by the time it gets here. It may not be threshold. But almost any one of these guys, unless they're just a super minimal signal, are going to reach threshold because they're right on that axon hillock. It's almost an instantaneous give, unless there's some inhibitor coming in, these are almost instantaneous fire off an axon. And that's where the real complexity ensues, is that we have this integration occurring in hundreds or thousands or tens of thousands of presynaptic terminals all firing off or not firing off at the same time. Imagine the complexity you could achieve with this level of integration. You can have truly extraordinary levels of connection. Neurotransmitters, ionotropic. Neuromodulators, metabotropic. This is a complex figure. Walk through each thing step by step. So let's start over here. We're going to get this channel and it causes a change in permeability. Let's say it's opening and it's letting in sodium. So now we've got more sodium. It causes a depolarization, and that's an excitatory postsynaptic potential. If the neurotransmitter is triggering another channel, like say a potassium channel out, or perhaps a chloride channel in, that potassium out or that chloride in is going to be inhibitory. There are possibilities where we can slow sodium leak, leading to hyperpolarization, or slow potassium leak, leading to depolarization. That's why knowing about these ions makes this figure easier. But walk through each possibility and say it in your head, say it out loud. Let your auditory side hear this in addition to thinking it. It will actually reinforce, or write it down in words, something along those lines, or you can even draw it out. Again, the act of drawing and reading the words and then writing the words, very, very powerful study tool. This is just an example of summation, and now we're talking about presynaptic terminals again. So we're kind of circling back here. 3 subhold signals simultaneously summate to create a threshold and act one or more action potentials. Or if we start to summate with pluses and minuses, maybe this minus being a little closer to the trigger zone is got more influence. So even though we happen to have 2 positives here, the fact that that negative is closer to that hillock means that it might be able to overcome. Or if these by themselves are sub-threshold, you knock some of that out and we're back to sub-threshold again. Just like an abacus, and in many ways it's a biological abacus. Temporal summation, for instance, if we're firing from the same location. So if we have 2 rapid fire pulses, we could reach threshold. And this is a picture. It's okay. I mean, it's certainly in color, which is nice. But it's showing you the synaptic. It's taking a branch of a dendrite, and it's showing you excitatory synaptic surfaces and inhibitory synaptic surfaces, and showing you that the integration occurs from the point we start receiving signals all the way down to the hillock, the number of action potentials we fire, the amount of neurotransmitter, and so forth and so forth. Remember how I showed you that picture of the cell body with all of the presynaptic terminals on it, like this one right here? Well, it turns out in terms of in neuroarchitecture, we get a similar type of structure relationship going on at the presynaptic level. So instead of it being another neuron, say right here, that is counteracting, it might actually be at the pre-secretion step, and that's what this picture is showing here. And what this can do is, let's say you want to have some kind of a response. You're firing off some sort of a signal, but you want to suppress a certain response. Meanwhile, other responses are going on. It allows for a coordinated activation and inhibition to meet a physiologic need. There may be times where, like for instance, if you stub your toe, that you don't want to, swear. Or there may be times when you're just by yourself and then you just let it go. So this guy doesn't fire off and you cuss a blue streak, if you know what I mean. But you're somewhere that's sensitive, so you don't want to be caught doing that. So other neurons in your brain suppress the neurotransmitter secretion. But going back here, that can occur this way at this integration step, or it can occur, let's take this blue and put it right over here, and we never secrete neurotransmitter. We don't have any here, but that would be what would happen. But the net result would be no response wherever the inhibition is occurring. We can have targeted inhibition, or we can have systemic inhibition. And the last few slides are just touching on neuromodulators a little bit. And I want to talk about this because many of you are psych majors or human behavior specialists. One of the really fascinating things is how we establish memory, how we develop behaviors and normalities and that sort of a thing in our neurocognitive system. And the leading, but not the only theory, but the leading theory is known as long-term potentiation or long-term depression. To make the story short, I'm going to show you this in the figure. When you have some signaling going on, you may have a given receptor population on that postsynaptic cell. But with the help of neuromodulators, after several of these stimuli and a little while, if you have repeated stimuli, you get more receptors. You become sensitive to it. I'm going to badly sing something here for you, and I'm going to ask you to fill in the blank. 8, 6, 7, 5, 3, 0, 9. It's a famous song from the 80s. Many people know of it or they've heard it from their parents or that sort of a thing. That recognition was due to the fact that you have more sensitive auditory pathways to that particular set of notes and that particular language. Long-term potentiation. is defined by sustained changes in the sensitivity, if you will, shall we say, the quality and quantity of connections. Yeah, you're going to establish new connections in some cases. Neurons that weren't associated before will form, but also neurons that are associated will also change the amount of receptor to which they are sensitive to a certain neurotransmitter. And so learning, memory, depression, and mental illness all seem to be related to this long-term potentiation and depression event. You think about post-TSD, certain stimuli can send people into triggers, and that's exactly where they have hypersensitive neural pathways. Here's some evidence for you. If we have just a synapse that we isolate from a model animal or so forth, if we fire off neurotransmitter, and you can do this through artificial electrode stimulation, you'll get a depolarization event. This one was actually measuring calcium in this particular experiment. Well, so we have a deflection, graded potential. Let's say we continue to stimulate repeatedly over a period of time. A week later, notice the change in the number of receptors, and the response is greater. That's what we're talking about with long-term potentiation. We have a much larger graded potential than we did just a few days earlier, and that's due to receptor expression, second message infrastructure, so forth. And here's the gentleman behind the long-term potentiation theory and much of the major aspects of neurophysiology that have survived close to a century or more. His name is Ramon Santiago y Cajal. He started in Spain as an apprentice to a barber. He was going to be a barber and then a shoemaker, a cobbler for a while, but it didn't stick. And he got interested in research and did an extraordinary amount of work with using just a very, very simple microscope that would almost be toy-like in today's terms. Very powerful stuff. And he drew these pictures, like for instance, this Purkinje cell in the inner part of the brain, or other areas, the layering of neurons in the cerebellum, and so forth. And he pioneered the ideas of long-term potentiation and neural patterning. They called him the Einstein of neurophysiology because his groundbreaking work just opened up doors for all to follow. In later years now, in the last 10 to 20 years, we have seen yet another watershed change in our understanding of how neurons work. Certainly, Ramon y Cajal, his theories and hypotheses hold, but we're finding out now that you can actually replace memories by selectively ablating neurons and then replacing them. What's amazing to me is this picture. Each of these neurons, each color is a different neurotransmitter. So you can see how complex it really gets. We can differentially stain neurons based on what their predominant neurotransmitter is, and you can see the extraordinary complexity. And we have just so much more to learn. It's absolutely fascinating, and it's growing by leaps and bounds. You've seen buildings go up here in Indianapolis dedicated to nothing but neurological research. And like I said, yes, the potentiation side of it is very, very important, but the patterning also seems to be important as well. That you can ablate a neuron in one of these pathways and then replace it with a new neuron, and the memory or the behavior returns. That's actually been done in mice. Pretty amazing stuff. That concludes Chapter 8. So we have a lot going on here, and that's why we have a whole week dedicated to this to processing this information, because it's a very, very big chapter. Unlike, say, last week where we had cell signaling and then endocrinology. So make sure you take your time with this. Note the words that we've used in the lectures and how they're being used here from the slides. And then the more you can use these words, the easier it is to start envisioning this in your head. Thanks again. Thanks for sticking around, and I will talk to you again soon.